Welcome back, friends. Let's talk about pot.com. I'm Primo, and I'm here with my new friend, Will Marrs, who is a grad student in neurobiology and behavior therapy here at UW in Seattle, right?
Sure.
He is close enough to being a doctor, doesn't like to be called a doctor, but I think it's close enough to qualify as one in my book, so I'm going to call him doctor. So, Dr. Will, nice to meet you.
All right, nice to meet you.
So what are we working on here?
I was asked to talk about our new research on enzyme of the endocannabinoid signaling system. We recently published this work in Nature Neuroscience, and so it's gotten a little bit of attention, and I thought I would just clear up exactly what we did.
Yeah, maybe we can take just a step back just for a minute and explain what the endocannabinoid system is.
Exactly.
When it was discovered, what we know about it, what we're trying to figure out, anything you can shed some light on for our viewers to understand. Because we've heard this word floating around, but no one really has a good grasp on what it's about.
Yeah, well, the reason it's called the endocannabinoid signaling system harkens back to its discovery, which had to do with cannabis, and that was in the 1960s when it was finally discovered that the psychoactive component of marijuana was actually THC, and that THC exerted its psychoactive effects by binding to a cannabinoid receptor, or what became called the cannabinoid CB1 receptor. And it turns out that we now know that the CB1 receptor is actually probably the most abundant receptor of its class, the G protein coupled receptors in the brain. And it's pretty much everywhere in the brain that you look for it. It's got higher expression levels than others, and it's not too highly expressed in the brain stem, but it's pretty much everywhere.
In other parts of the body too, right?
Yeah, it's throughout the body. I mean, my focus is on the brain, but I think similar mechanisms are going on, and it's certainly highly expressed throughout the body.
So there are molecules like cannabis molecules already in our body before we even have contact with cannabis from the outside.
Yeah, that's right. So it was kind of funny in hindsight. At the time they discovered this receptor that binds THC, and that's how people get high, but we didn't know why we had that receptor. So for a while, people were saying, oh, well, you know, God wanted us to get high, he gave us this receptor. But it turns out that it was discovered that there are actually naturally produced chemicals that do bind the cannabinoid receptors, and those became known as endocannabinoids. The first one was anandamide, and the second one to be discovered, which is what I've primarily studied, is 2-arachidonylglycerol, or 2-AG. And that one turns out to be more of a prototypical endocannabinoid, at least when looking at it from a neuroscience perspective, in the sense that it's a full agonist at the cannabinoid receptors. It's produced at the synapse where we would expect it to be produced and activated right near the CB1 receptor by an enzyme called monoacylglycerol lipase, or MAG lipase. And if I'm not going too fast, that's actually where I get into the story, because we found another enzyme, this ABHD6, which is very similar to MAG lipase. It also inactivates 2-AG and thereby controls the level of 2-AG, and indirectly thereby controls CB1 receptor activation.
It's this whole control system thing that I keep getting when I read about it. It can push things up or slow them down depending on...
Yeah. It's been called an on-demand signaling system. So if we consider the synapse in the brain where two brain cells come together, the classical method of synaptic transmission is the neurotransmitters are released from the presynaptic neuron, or neuron number 1, travel across the synaptic cleft to activate the receptors on the postsynaptic neuron, or neuron number 2. And that's how information flows in the brain, and that's very well characterized. But the endocannabinoid signaling system turns out to be an example, and there are others, but we now know a very important example of a retrograde signaling system where it goes from the postsynaptic neuron, neuron number 2, back to presynaptic. And it's an important process to allow the postsynaptic neuron to regulate the information that it's getting, basically feedback and say, this is my status. And it's found in synapses throughout the brain. Like I said, it's just sort of a universal mechanism for synaptic regulation. And so it's called on-demand because the endocannabinoids, and 2-AG in particular, is produced only really when the postsynaptic neuron is overactive. So it's getting a lot of excitatory neurotransmitter, a lot of calcium is coming into the cell, and it's basically starting to say, okay, enough is enough. The molecular machinery gets kicked into place. 2-AG is produced from from lipid, basically from the membrane, from molecules that are actually there in the fatty membrane right there. And then the 2-AG is produced and is thought to travel retrogradely to the presynaptic neuron, where that's where the CB1 receptor is located. And then when you activate the cannabinoid receptor, excuse me, it actually sends a signal to decrease the release of the neurotransmitter. And we know that the 2-AG is then degraded, and that degradation is what modulates the level of the signal, and that's going to control how much CB1 is activated and everything else. And so this MAG lipase enzyme, which was previously already known, is located right there presynaptically near the CB1 receptor, so kind of at the end of the signal. And then it degrades it, and there's this beautiful molecular logic there that people have been talking about. And that's kind of where our story got started.
How did you get involved in the study? How did your interest in this?
Well, I've always been interested in neuroscience. I'm in the neurobiology program here. More recently, I've been particularly interested in the endocannabinoid system. I've been here in Dr. Nefistella's lab for 4 years now, studying the endocannabinoid signaling system. And I actually got started. It turned out to be kind of a lot of serendipity the way the project unfolded, but I got started on a microglial cell line. The microglial cells are the immune cells in the brain. They're not very well appreciated. Most people study neurons. But there are other cells in the brain, glial cells, and one specific type of glial cell is this immune cell, the microglia, and that's a hot topic in the cell lab study. So I got started on a cell line, just not even from an animal, but just a cell line growing in the lab, and a postdoc that was leading when I got started had discovered that this cell line had the enzymatic ability to degrade 2-AG, the endocannabinoid, but it did not express the enzyme MAG lipase. So I basically inherited this story where we have a cell that has an enzymatic activity with an unknown enzyme mediating it. And so to cut to the chase, we, through a collaboration with the Curlat lab down in San Diego, who has this really efficient protein profiling technique called activity-based protein profiling, we sent them samples from our cells, and they were able to send us back potential candidates in the serine hydrolase enzyme family, which we had reason to believe is what this mysterious enzyme was. That gave us a few candidates. We followed up and determined that the enzyme that was degrading 2-AG in these cells was actually ABHD6. And that's a mouthful. It stands for alpha-beta hydrolase domain containing protein number 6. But ABHD6, maybe it'll get named something else one day, but for now, that's it. And so that was where it started. Then we found this enzyme, and I started to look at neurons. I really wanted to get back to neurons and back to neuroscience. And so we looked at neurons. It turned out it was highly expressed in neurons, and that it's also highly expressed in the brain, and that it's doing everything we thought it might be doing, similar to MAG lipase, controlling 2-AG levels. And in fact, if you can inhibit this enzyme ABHD6 at a synapse in a brain slice from a mouse, in the cortex, you can control the synaptic plasticity through CB1 as we would expect. So it's expressed, it's functional, and it's an active part of the endocannabinoid signal.
And the potential payoff in real illness is?
Well, it's hard to speculate. So we do have precedent. I said MAG lipase was an enzyme that was known. Another enzyme is FA, fatty acid amide hydrolase, which hydrolyzes the other endocannabinoid, the first to be discovered, anandamide. And that enzyme, FA, is actually very well characterized and has been targeted therapeutically with great success. And if my understanding is correct, I believe that some of these inhibitors of FA might be in clinical trials or reaching clinical trials. I'm not sure, but they've had good success in animal models for ameliorating pain. They've shown some antidepressive, anti-anxiety